Okay, so we have previously prepared this data set. Basically, what we did was actually integrated some of the clinical data, including the treatment, the drug that's being used for the treatment, as well as the clinical response as well. We also remove some of the uh, information from the patients that are not being treated with gemcitabine, which is not part of the study, and end up with this data set here. Now, in this exercise, what I'm going to do is I'm going to show you how to input this data into a software, what we call glucomic, which is a very comprehensive and user-friendly software for you to do simple bioinformatics. And in this case here, we will be doing uh, supervised clustering or classification using three different methods, okay, kNN, SVM, as well as random tree to identify the gene signature that is associated with gemcitabine response. Now, to do that, first I need to reformat this data sheet into the format that it could recognize. Basically, it could recognize most of the spreadsheet data. It just need to be formatted accordingly. So I'm going to insert one column here. So basically, then I'm going to insert another row here. So what I need to do is basically now this this information here will be referred to this part, and then here I will have the microRNA uh, denomination. Create another title here. So now you basically have a 2 by 2 table. The matrix that you can actually input it in. Now, so the one thing is that for glucomic, it cannot recognize the Excel format. In fact, most of the program they would not recognize it. So what I'm going to do is I'm going to save it as comma delimited text file. So okay, I'm going to save this here, and yes. Okay, so now if you go to the data set here, you should have a text file like this. So next, what I'm going to do is going to open the glucomics. So this is the interface. So you can actually download this software, but you only have a 30-day trial license if you want to use it to try it out. Okay, so now here I'm going to insert the data set that I have just created just now, so which will be under research, and also the new folder. Okay, so now make sure that you actually include, well, you actually open the comma delimited file. So here you go. You can use the wizard to import the information. Now, the good thing about using the file import wizard is that it will run you through the step to step, so that you won't you won't actually make a mistake. Okay, so here to ask you select separator. The separator is comma, so it's selected quite well. Okay, the rows will be as each rows will be as a variable, and then now it will tell you, okay, data matrix. Okay, select the top left cell. Okay, so you basically uh, so you select this is the data set. This is the data that you have. Then next, it will ask you to select where the range. Okay, now you have matched. The blue color is where the data is 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 located, and then now the row, the variable. So this will be your variables, and then this will be your sample. Okay, annotation. Okay, so then you can click finish like this. So immediately, okay, so now it can actually give you a very nice PCA analysis as well if you want to use it. So okay, here what I'm going to do is, remember we have already annotated whether the patient is a responder or non-responder under this headline here, response. So I'm going to select that. So now you have two group here. So what I'm going to do here is I'm going to put the sample size. Now you can see that each dot basically represents each patient, and this is how the information uh, the microRNA variable information when you put it together, so how they segregated inside the in this population. Now note that this is a raw data. We haven't actually filtered out anything yet, okay, and we haven't actually identified the markers at this point. Now the next thing, as I mentioned before, you can actually include statistics analysis here if you want to. Okay, so that, well, you can actually filter out some of the noise. So maybe what I would do is I'm going to try out, okay, a two-group comparison. So by looking at responder versus non-responder, and then you can change the p-value to 0.05, let's say. So now then, basically, what you have here is now you actually filter out those that are not statistical significant. So whatever is remained is the one that is statistical significant. So we can use that if you want. You can also include the fold change, okay? See how many different fold change in terms of mean to see whether they, okay, can separate quite nicely or not. In this case here, I think the fold change might not help you that much, okay? But anyway, so, okay, so this is how we can actually do the filter. Now, so the next thing what I'm going to do is I'm going to build the classifier here. Now, before I do that, I'm going to show you, and there's a few other plot that you can look at. Another way is a heat map. So you've probably seen this quite a lot, okay, from publication papers and so forth. So it's actually a good way to look at it as well. You can read out how the heat map look like. Of course, here it is now random, basically. But if I'm going to change it to, let's say, the variable is miRNA. So if I'm going to change it to here, okay, the okay, I'm actually do a hierarchical clustering here, okay, and also then the sample I can actually do put it as we call it the sample color, okay, based on responder, okay. Okay, so now you can see that there are still some of the column with a lot of zero here, but nevertheless, okay, so we can explore a little bit more about the heat map, okay, so you can actually show quite a good separation sometimes, okay, for example, to test whether the data set can be I mean, your microRNA signature can be separated into two based on the response or not. You can also do hierarchical clustering on both sides. So now you can see that, okay, you have some group of this. Oops, I can probably none here. Oh, put response. Okay, okay, here you go. So now how to read this map is basically, okay, you have a group of patients that have downregulated of this microRNA, and some of these are correlated with the yellow, which is a responder. But again, the separation is actually not that great. So if the separation is good, you can get a quite clear cut whereby most of the blue will be on one side and most of the yellow will be on another side. Again, so this basically show you is that, well, if you are just going to rely on the statistic and t-test, well, this is the result that you will get. That's very unlikely that you can get a clear separation. So now to move forward, what I'm going to do now is I'm going to actually build the using the classification methods here. So I'm going to build the classifier. In this case here, I'm going to use kNN. Okay, I'm just going to change it to two group comparison since we are doing responder versus non-response. So, and the key would be based on the response. So what I'm going to do now, so I do not have the validation set at this point, or if I want, I can use the same set to do the validation as well. Okay, so now what I'm going to do now, I'm just going to simply click the build button here. So it will give you a warning, but go ahead. So you can save the data set here. So it will take a while to build it. So now that's it. It's done. So it's built basically. So what it show you here is basically using the kNN. So the accuracy of the non-responder is about 0.89%, and the recognition, the accuracy for the responder is 0.84%. What it means is that, well, and if you scroll it down, okay, you'll get a number of this microRNA here. This is the so-called gene signature that, I mean, or the microRNA signature in this case here, that can be used to identify the responder versus the non-responder. And in this case here, if you just look at these few markers, then you can actually predict whether the individual is responder or non-responder. In this case here, the non-responder prediction accuracy is 0.89, which is 89%, and also the responder is 84%, which is considered actually quite good. So, well, you can actually repeat the same what you call it, the same analysis by using different methods. Okay, let's see if let's export this information out. Okay. Okay, so this is the kNN information. So you can also equally use another method, SVM, for example, and you just click build again. So okay, then you save it. So then here you go. If you are using SVM, so now you get more markers that the algorithm have identified could predict the response. And in this case here, you get almost 100% now. Be careful with SVM is that sometimes SVM tends to overfit the data, but you can read up more about it. But basically, well, you just have to be careful not to overfitting. That's why in your study, it's good actually to use 3 different methods. Then later on, you can compare and validate the data later on. So at this stage here, I need to emphasize is that this is just a preliminary building of the information only. So you can export this data again. So this will be under SVM. Okay, can save this. Now, similarly, you can also build using the random tree again. Well, you just press this button here. Okay, 40. So random tree will take a little bit longer time. Depends on your processing power. Sometimes it could take a much longer time. But this is a relatively small data set. Imagine if you are going to put in the mRNA expression, it will take some time to actually build. So just be patient about it. Okay, here you go. Under random tree, you actually now have quite a lot of markers here. Okay, and then the accuracy again is about 100%. So again, this is basically building the model using the same data set and doing the validation using the same data set. So it's not unusual that you will get a high accuracy, but that's why the independent data set will be very important to actually use it. Okay, so I think that is all for now. So thank you.